Hello to everybody. Good morning, good afternoon, good evening, wherever you are. And this is the session 4 that I'm opening together with Walid Haney, that we are co-chairing this afternoon here in Italy. I am Gabriella Scarlatti from Milano at the Institute of San Raffaele. And the session is Animal Models for PrEP and Vaccines. And we have 2 fantastic presentations plus 2 other talks that were selected from the abstract submission. And so I have the pleasure to introduce the first speaker, Gerardo García-Lerma. Gerardo is a team leader at the Antiretroviral Prophylaxis unit at the CDC, and he will talk about modeling, macaque modeling of antiretroviral prophylaxis for HIV prevention, and has been one of his major fields of research since years, besides many other things. Thank you, Gerardo.
Hello, thank you for inviting me to give this presentation. I'm going to be talking today about macaque modeling of antiretroviral prophylaxis for HIV prevention. These are my conflicts of interest. Why is important to use, why NHP or non-human-primate models are important to assess the efficacy? First, these models can provide proof of concept of efficacy. They can measure biological efficacy versus effectiveness, since we are able to control drug dosing. We can investigate relations between efficacy and pharmacological parameters, drug concentration, for systemically or in tissues. We can inform dose selection and regimen modality for clinical trials. And the models can help prioritize PrEP products, which may result in substantial savings, since phase 2B and 3 trials are very expensive. There are 2 key considerations for assessing PrEP strategies in NHP models. First, we need to consider pharmacological aspects. It is important that we model clinically relevant drug doses. And if we don't do that, we need to understand what is the translation of those doses. We need to adjust doses based on the difference in size and metabolism between macaques and humans, which is called allometric scaling. And we should incorporate drug measurements at the mucosal site of infection. And this is one of the advantages of this model that led us to collect specimens in multiple locations and understand drug concentrations of how they relate to infection. And then we have the transmission component that is equally important. The models use physiological viral inoculum that mimics HIV infection doses in humans. We need to make sure that we select appropriate species. For instance, pigtail macaques are better suited for vaginal transmission studies or prevention studies. We use an R5 tropic sheaf isolate that mimics HIV-transmitted viruses. And then one key characteristic is the repeated low dose virus exposure nature of this model. So when we combine the pharmacological aspects and the transmission component, we end up with a model that is highly relevant to PrEP studies. The repeat low dose models that were developed at Cecilio in early 2000s have predicted efficacy of Truvada for PrEP. This is the first work that we did with this model. And the table shows the specific PrEP regimen that we investigated, the biological efficacy that was identified in macaques, but different through different routes of exposure, and the efficacy. As just an example, when we look at the daily FTC-TDF, in the macaque model, the efficacy was 87% against rectal infection. And when we look at efficacy in humans with detectable drug, we can see that the efficacy goes between 90% in the public spread to 100% in the PROUD study. So, and the same happened with on-demand FTC-TDF, where we model rectal efficacy in the macaque model with a 2+1+1 dose, found 94 to 100% efficacy. And the IPERGAY later showed that the efficacy of this intervention was approximately 100%. We not only use efficacy, but we can inform on potential modifiers of the efficacy. We've done many studies with FTC-TDF through the years, and we've shown that, for instance, the efficacy of this combination is not affected by the injectable contraceptive we found a very modest reduction of efficacy in macaques co-infected with chlamydia and trichomonas. We demonstrated high efficacy of FTC-TDF against FTC-resistant virus containing the 184 mutation, and we also found partial efficacy against the non-resistant that have K65R. These are very important findings because K65R and A184B are two of the most prevalent mutations associated with acquired drug resistance. So if we look at the prevention spectrum, what we have currently available, as I showed earlier, we've been able to provide proof of concept of daily oral PrEP, FTC-TDF, as well as on-demand PrEP. But we have also investigated products that are in development and have efficacy trials underway. The clinical work has been able to provide concept for many of these interventions. And I want to focus on oral PrEP with FTC-TAF and the long-acting cabotegravir. The first study that provided proof of concept of long-acting cabotegravir was done at the Roundtable Research Institute. And this study investigated the ability of injections of cabotegravir that mimic human doses to protect macaques against rectal virus exposure. The study showed high efficacy, but not only that, was able to identify correlates of protection. In this case, when the concentration of cabotegravir were above 4 times the protein adjusted by C90, the level of protection was 100%. And when it fell to between 1 and 3, the level of protection still was high, around 97%. So not only protection, but also correlates of protection. A more recent study published by our group also investigated the efficacy of cabotegravir using a newly developed repeated denial shift transmission model, and found that cabotegravir was as effective as FTC-TDF in the model. There was one breakthrough infection in the cabotegravir group, but that animal had concentration of cabotegravir in plasma that were below the protective threshold. I know we all heard the results of the HPTN-083 presented a few months ago that showed that PrEP with cabotegravir long-acting was superior to FTC-TDF among MSM and transgender women, and this resulted in a 66% reduction in increase of infection with cabotegravir compared to FTC. So again, the clinical model using the rectal and vaginal transmission model was able to predict the efficacy of cabotegravir long-acting. There are 2 more studies that have done with cabotegravir to investigate efficacy against vaginal challenge. In one study published in our group, we showed that cabotegravir was protective in macaques that were repeatedly exposed to low doses of SHIP. And a second study that done at the Iron Diamond showed that cabotegravir was also highly effective in macaques in a different species, in rhesus macaque in this case, that were treated with injectable contraceptive DMT. And in this case, there was also high level of protection. And we heard a few days ago that the HPTN-084 reported very good news. This study was originally expected to continue to 2022. The DSMB that met in November 5th, 2020, found that cabotegravir long-acting was superior to daily oral FTC. So expanding the results that were reported for rectal protection out to vaginal protection. All findings were able predicted by the clinical study. The next agent that is under clinical trials is oral FTC-TAF, and these are the studies that we published with oral FTC-TAF. First study that we published in 2016 showed 100% protection when animals were receiving this combination against rectal transmission. And a second study that we just published a few months ago showed that the combination of the FTC-TAF was also highly effective in infection. In this case, we have a 1 breakthrough infection in the study, as shown in the survival analysis of this breakthrough infection was again associated with the little or no detectable phenotype, which sometimes has happened in pigtail macaques. For some reason, they may not metabolize the drug very much. We know data on FTC-TAF from the Discover trial that was presented at PRO in 2019 and published months ago, showing that FTC TAF was as effective as CPF, again, finding that was predicted by the rectal macaque study. We still don't have data in cisgender woman. This is something that is ongoing, so we hope that this same combination is also will also be high. So I've described a couple of products that are now in development based on very encouraging technical studies in primates. And I'm going to now move into products that are at various preclinical and clinical stages at this point. And I will touch base on some of these products. The first product that is very promising is islatravir, or EFDA, also known as 8591. This is a first-in-class nucleoside reverse translocation inhibitor that has three very unique molecular structural components that make it reverse transcriptase inhibitor, translocation inhibitor, and provides a very long half-life. The first study that was done in rhesus macaque was very encouraging, and so that one single weekly dose of this molecule was able to protect macaques against repeated virus exposure. This was a result that was published a year ago, but the Diamond Group, again, very encouraging data. We've been interested in new oral regimens that may improve on-demand modalities over existing on-demand regimens. So we have been interested in evaluating a single oral dose that we use either for PrEP or for PrEP. So we did a willingness evaluation of this modality among MSM in the US and found that the preference was there for a single dose, either PrEP or PrEP compared to PrEP. So we investigated in macaques the biological efficacy of this single dose PrEP modality. And the rationale is that now we have much better drugs than when we started discussing about PrEP. We have TAF that has a long persistent experience in that issue. So in principle, could provide extended duration of drug exposure. And we also have integrase inhibitor that act at later stages of the pediatric cycle and therefore are attractive candidates for. So we designed a series of virus challenge experiments where we administer macaques one single dose of PrEP, either 24 hours or 4 hours before virus challenge, the top panel, and found that these modalities were highly effective in preventing infection. And we also defined this same combination as PrEP, administering the doses from 2 to 48 hours after virus challenges. And we saw high efficacy, but definitely the more you wait to administer the PrEP dose, the less efficacy you will have. So we demonstrated time response with this intervention. This was a combination that's very promising that included C, as I mentioned, TAF and the integrase inhibitor More products in the pipeline, very promising. These are in situ forming implants that have been developed by the Universitario group at UNC. These implants basically form a depot after administration, and then they are slowly degrade and release antiretroviral drugs. Very interesting technology that is currently in evaluation. We've been collaborating with them and show that this drug delivery system is able to sustain drug concentrations. In this case of dolutegravir, for up to 140 days. So very sustained release. Another implant that is very promising is a biodegradable implant in TAF that has been developed by RTI International. And again, we've been doing some preclinical evaluation of this implant. And this is again another biodegradable implant, and the graph shows sustained concentrations of tenofovir diphosphate PBMCs with 3 different implants containing low, mid, or high dose for between 12 and 20 weeks of study follow-up. So again, very high levels of tenofovir diphosphate above what has been seen with daily oral TAF and sustained for several months. So very promising results. Another promising technology are intravaginal rings. In this case, are 3D printed rings. These have been developed by Rahima, also at UNC. These rings are used for some multi-purpose prevention technology against HIV, HFP, and contraceptive, pritelivir, LNG, and rilpivirine. So again, very promising strategy where you can add not only anti-HIV drugs, but also anti-HFP and contraceptives. Another modality that is being currently under evaluation in non-human primates are vaginal inserts containing TAP-G for on-demand PrEP or PEP. This is work done in collaboration with Conrad to develop the product. We've been investigating in non-human primates the efficacy of these inserts, either as PrEP or PEP, and the results are very encouraging, so high level of protection against vaginal infection. So in summary, the repeat low-dose macaque models of rectal, vaginal, and penile shift transmission has been used to provide proof of concept of daily, on-demand, and long-acting PrEP. The models have been validated by clinical trial results with FTCTF, FTCTAP, and cabotegravir long-acting. Has been highly predictive, very high predictive value. 7 of the 7 trials have been efficacy has been predicted by the repeat low-dose model, and can not only inform on efficacy, but can also evaluate potential modifiers of PrEP efficacy under highly controlled conditions, exposure to drug-resistant viruses or situations that might be such as co-infection with other sexually transmitted infections. The repeat low-dose models is also being used to identify promising second, third generation products. This includes, as I described briefly, novel oral antiretroviral drugs or modalities, biodegradables and removable implants, intravaginal rings, inserts, and of course, multi-purpose prevention technology that not only against HIV, but for other experiments. Finally, I want to acknowledge everybody that has part or is part of this effort. I want to specifically acknowledge the antiretroviral prophylaxis team. The systemic prophylaxis activity is led by Chan Tovar, Ivana, Miller, Michelle, and Kenji, and then the topical prophylaxis activity led by Smith, Natalia, Tiana, and Don. Other teams of the branch other than Mike has also been involved in this effort, the clinical evaluation team, the analytical and clinical evaluation team, and the diagnostic team, the statistical team, and of course, all the collaborations that are working with us on this. Thank you very much.
Hi. So that was a very, very interesting talk by Gerardo, and that showed how informative macaque models can be for going into human trials, and much more than that. Now I have the pleasure to introduce the next speaker, David Masopust. He is a distinguished professor at the University of Minnesota Medical School, and he will talk about the potential role for tissue-resident memory T cells in a protective vaccine against HIV. I want also to remind everybody that you should pose all your questions in the chat box so that we will, at the end, we will have the session and we will be able to have a lively discussion, and you can get replies to all your doubts and questions. Please do that. So please, David, go along.
Today, I'll be talking to you about the potential role for tissue-resident memory T cells in a protective vaccine against HIV. And I have nothing to disclose. So I'll begin with a familiar problem, and that is that the transmission efficiency of HIV across the mucosal barrier may be low, meaning that only a few founder cells are productively infected. But before the adaptive immune system can engage, we have to wait for viral amplification to provide antigens that get to the draining lymph node to activate those rare naive CD8 T cells or B cells to undergo clonal expansion, to be numerically relevant, to acquire the appropriate effector functions, and then in the case of CD8 T cells, to migrate to the site of the action. And so this primary response is slow, and viral replication is fast, too fast, and the founder effect opportunity is certainly not taken advantage of in a primary immune response. So in the vaccine setting, we have to change this, have to establish a pre-existing immunity that would result in a sufficient quantity of lymphocytes that are in the right locations to recognize antigen really at the portal of entry, why the infection is still a small problem. And those cells would have to express the relevant functions or the right quality. And from my perspective, HIV vaccine efforts ongoing today are largely tilted towards generating productive antibody responses, including the elusive broadly neutralizing antibody. And I agree that's a very laudable goal, and I support it fully. But there's relatively little on the CD8 T-cell vaccine side going on today, maybe most notably Lewis Picker's work with the CMV vector. But clearly most ongoing vaccine efforts are not attempting to potently utilize both arms of the immune system. And I'm going to argue that maybe we should. And so I'll begin by just giving you a very brief refresher course on T-cell immunosurveillance. So naive T-cells are surveying a very small part of the organism, namely the secondary lymphoid organs such as lymph nodes, and they're doing so through a program of recirculation using blood and lymph as a conduit. And so in the event that you get an infection at a barrier surface, I've drawn that here in the big toe, those T cells, like I said earlier, have to wait for antigen to come to them, to be stimulated, to proliferate, to become numerically relevant, to acquire effector functions. And then because they're T cells, which are tactile, they have to migrate out into non-lymphoid tissues and survey host cells and ask if they're expressing their cognate antigen or if they're infected. In this case, the answer is yes, and has resulted in a prototypic function of CD8 T cells, and that is cytolysis of the target. Importantly, after a vaccine or an acute viral infection, that expanded population of memory T cells that remains to fight another day is now more broadly distributed. So like their naive counterparts, they can be isolated from lymph node or blood, but they're also present in non-lymphoid tissues, including mucosal barrier sites. And those CD8 T cells in non-lymphoid tissues, by and large, are something that we call residents. And what I mean by that is during a primary immune response, when you have naive cells that get activated in a lymph node, they proliferate, and some migrate out into a non-lymphoid tissue, the female reproductive tract, for instance, well, those cells are going to undergo an in situ differentiation program that results in those cells becoming parked within the tissue, or what we call resident. And we call these cells TRM for short, for T resident memory. And I want to emphasize, this is probably the major population of T cells in your body. They are phenotypically and functionally and transcriptionally distinct from the kinds of T cells that you will find in blood. And this was not appreciated until relatively recently. These cells in blood, by and large, are not surveying these frontline tissues, these mucosal tissues. That's left to the residents that were established during the effector phase of an immune response. And so I've been studying these resident memory T cells for the last 20 years. This is intravital microscopy. This is the female reproductive tract of a living mouse. And even though these cells are resident, they're moving, and they're cells looking for evidence of reinfection. And so what we've done is we've created mice where we could visualize separately both CD8 T cells and TRM in the female reproductive tract, along with CD4 T cells. And we asked, what is the efficiency of immune surveillance, at least in a mouse as a starting point? Meaning, if you wanted to find a rare founder cell, how big would your army of CD8 T cells have to be to scan every potential target, every CD4 T cell in that tissue within a given time frame? Hard data based on in vivo analysis. And the answer is this, is if we have a large TRM army in the female reproductive tract, they could scan every CD4 T cell within 3 hours. Perhaps we could do better with a larger army. But if we had a small army, it's quite remarkable, but we're on the left end of this curve, 21 days. And this army is the extrapolated prediction of what was provided by the STEP vaccine trial. This was a T-cell vaccine that went into humans and failed to protect against HIV. And so, maybe this is a back of the envelope calculation. It's dependent on mouse data, but clearly 21 days is way too slow to find those founder cells while they're still rare before the virus is amplified. So we believe that you need to get to this end of the curve. We've made that prediction. We've done a lot of work to see how we could establish large quantities of memory CD8 T cells in mucosal tissues. And I'll share one with you, the strategy of heterologous prime boost vaccination. And so what do I mean by this? Well, we're using immunogenic vectors, and we're giving 3 independent immunizations. Each vector shares something that we want the CD8 T cells to respond to, but they avoid cross-reactive neutralizing antibodies. So we're avoiding interference with other aspects of the immune system to concentrate the immunogenicity of each immunization on the CD8 T cells themselves. And I'll just show you what this looks like. So we've primed a mouse. We're looking at our response to the vaccine with a tetramer. These are memory cells in blood. About 1% of CD8 T cells are vaccine specific, which is pretty good. But we can boost, that goes up to 8%. And if we give 3 immunizations, astonishingly, we have a durable memory that comprises 60% of the CD8 T cells in the blood of these mice. And this just shows what is really possible within the mammalian organism. And I won't show you all the data, but abundant memory CD8 T cells are really present throughout the organism and in non-lymphoid tissues as well. And so we asked, could we translate this into rhesus macaques? And so we took a similar vaccine modality. It's a VSV, a vaccinia, and an Ad5 vector containing full-length SIV gag in this case. And we're immunizing rhesus macaques that are MAMO-A1 positive. And what that means is we can track the immunodominant response to gag with a single MHC class 1 tetramer, referred to as CM9 here. And so if we prime monkeys, we get a response, we get a memory. It's pretty small, 0.1% of CD8 T cells, not as good as the mice. We can boost, and we can boost again, and now you're getting upwards of, in many animals, 5% or even 10% of the CD8 T cells being memory T cells that are specific for gag. Again, it's not as good as the mice, and we think we know why. We think we could do better, but nevertheless, we proceeded forward with the vector modality that we had. I will mention that these cells, the memory cells, are broadly distributed. We've looked at about 25 different tissues. I'll emphasize the female reproductive tract because that will be the site of challenge. So we have abundant memory populations that are gag specific in this tissue, and by phenotypic criteria, they look like resident memory T cells. And in fact, you can visualize them with immunohistochemistry and in situ tetramer staining. So every red cell is a SIV gag specific cell in the vaginal mucosa of an animal that was vaccinated 294 days ago. So what we've done here is we've loaded this frontline portal of entry with pre-existing SIV gag specific CD8 T cells. So in the event of challenge, there will already be cells on site, and many of them patrolling to hopefully find a rare found or infected cell and hopefully to do something about it before it propagates the virus to neighboring cells. So we challenge these animals with a high dose of SIV MAC 251. It's a swarm that's related to SIV MAC 239. And again, it's potently pathogenic, and it's a high dose. And our naive animals acquired the infection with a single challenge. But our vaccinated animals, what we observed was that 5 of the 8 animals, we failed to detect viremia. 3 animals were viremic, but there was evidence that the viremia was delayed. We couldn't see that viremia until longer after challenge, and then the peak viremia was lower as well. So this suggested to us that it was plausible that because there's only a gag insert, there's not going to be relevant antibody here, so that CD8 T cells at the front lines were capable of early recognition and early some sort of effector function that contained or suppressed the infection or perhaps eliminated it at the mucosal barrier. And what was interesting, it's a hypothesis of that interpretation, is if you looked at the difference between the animals that had putative protection versus those that did not, well, they were different in terms of the magnitude of the CD8 T cells that had been established that were gag specific. Now, this was only measured in blood. We didn't want to sample the mucosa before challenging that mucosa. But it seemed to support the hypothesis going in that there was a threshold. And unfortunately, that threshold would be, somewhere upwards of 5% of the CD8 T cells in blood, correlating with protective efficacy in the mucosa, but relating to magnitude. And so we did not publish these data, and it's actually a few years old, and because we have an N of 8, and that wasn't enough to make me comfortable putting weighty conclusions on. So I shared that with you, though, for context and for you to make your own interpretations. But fast forward a few years, and this is a happy collaboration with Emory and with Eric Hunter, Cynthia Dearden, Rama Amara, and then Bali Palendran. Then the paper came out in Nature Medicine, and what we had done was combine antibody and CD8 T cell vaccine concepts. So we're trying to bring the two together. And so I won't go over the granular details. It's a published report, but we had a control group of animals. We had a cohort of animals that got a native SOSIP trimer immunization adjuvanted with a TLR7 agonist, 3MO52 here. And what this does is this elicits neutralizing antibodies that will be effective against an autologous SHIV challenge, which is what's going to be done here. And so if any animal that has a high enough antibody titer will be protected from infection. And that titer is over 300. We had a separate group where we didn't use CD8 T cells alone, but we had the heterologous prime boost vaccination to elicit CD8 T cells, and we also included the protein immunization to generate envelope-specific antibodies to see what the two might do together. And I do want to highlight that the magnitude of the memory CD8 T cell population that was achieved this time around with this vaccination was not as big as what I showed you before, unfortunately. And we really didn't have any animals that exceeded our predicted threshold value of protection. But that doesn't matter because we also have antibodies on board. And so when we challenge the animals, again, if anyone had a titer of over 300, they were protected. And this is quite similar to reports out of Dennis Burton's group with a rectal viral challenge. But low titers were not protective as anticipated. But if we had included CD8 T cells or a CD8 T cell vaccine into those animals, now we saw an enhanced degree of protection, interestingly. And these would be GAG-specific CD8s. And what was, I think even more compelling to me is that if we waited another 140 days after our initial round of low-dose repeat challenges had ended, well, the antibody titers unfortunately waned in all animals, as they tend to do after this vaccine modality. And so the protective titer was lost. And in fact, if you challenged animals again that had only gotten an antibody-based sort of immune response, then they were vulnerable. But if we had included CD8 T cells in our vaccine regimen, then even at this durable time point, you still maintained evidence of protective immunity. So, it's suggestive that CD8 T cells might be relevant to include. How might CD8 T cells contribute to protection? What is actually the function of TRM? It's a relatively newly appreciated T cell population. Hasn't been looked at nearly as well. So they do kill. The green cell is a CD8 T cell. This is the female reproductive tract, and it's killing that purple CD4 T cell. And that killing occurs within the tissue within about 3 hours, even though they're memory cells, they are poised to kill. But a lot of other biology occurs when TRM recognize their cognate antigen. And so we've had a series of many papers and ongoing work on this, other groups as well, so finding similar conclusions that when TRM recognize antigen in tissue, they make cytokines. And those cytokines activate the local neighborhood, and they induce ISGs in an antiviral state. They activate the innate immune system and NK cells and dendritic cells. They trigger chemokines or VCAM1 in the vascular endothelium. So those cells in the blood, B cells and T cells that normally are not surveying this compartment directly in the steady state, are invited in and are recruited and migrated in as a consequence of CD8 T cell activation. And this is a very local response. And we now have a significant unpublished data that I won't share today of transudation of antibody. So if a CD8 T cell gets activated, it will bring in antibody from the blood. This is just one old data point here defending the conclusion that B cells are recruited. Here we are in the steady state in the female reproductive tract. If we've reactivated TRM, you recruit a significant number of B cells. So the point I want to emphasize is that TRM express their full range of functions in intact tissues. If you create a single cell suspension, you're going to miss out on most of the biology because the biology is dependent on the context of being in a neighborhood with other cell types, and they communicate with each other. They talk to each other. This is how TRM work. They live in tissues, and they respond, and they communicate, and they direct that environment. So how do we test this function in non-human primates? It's much more difficult than a mouse. We have developed ex vivo explant culture systems to study TRM biology. I'm not going to show you that today because it's a very short talk, but I'll share a few quick observations where what we've done is actually treated vaccinated rhesus macaques with Depo-provera in the epithelium, and then delivered cognate peptide into the vagina. And what happens is you get basically direct presentation among endogenous MHC class 1 within the tissue. And we've worked all this out and shown that that's sufficient to get a local TRM reactivation. And then we've done a battery of assays. There's a huge amount of data. I'll just show you a tiny bit. One tool we've used is CytSeq, which is using barcoded antibodies to identify specific cell types and marry that to single cell RNA sequencing to look at gene expression. And so by example, we can identify fibroblasts or endothelial cells, and those would be here in these UMAPs. The baseline, they're blue, meaning they're not expressing, in this case, CXCL9 or CXCL10, but very shortly after TRM reactivation, these populations of cells have upregulated these chemokines, which in turn recruit cells. And there's just myriad changes after local CD8 T cell reactivation. There's changes in the CD8 compartment in terms of the gene expression, but also the representative subsets that are there within the female reproductive tract. We see the appearance of NK cells, more activated ILC1s, and then a lot of changes within the B cell compartment, which we've putatively identified as B1 cells and memory B cells, although work remains there. And what we believe to be happening based on 3D clarity and microscopy or other imaging approaches is that we're recruiting a lot of B cells and we're recruiting a lot of T cells, just like we've seen in the mouse. And so a summary of what we know so far, we believe we have rapid and robust T and B cell recruitment to the FRT after CD8 TRM reactivation. We're characterizing the B cells more carefully. A mixed blessing, we are getting some CD4 recruitment, which of course are an HIV target. They are low for CCR5 for what it's worth, and interestingly, they've markedly upregulated HIV restriction factors such as ApoBec. Chemokines are upregulated, VCAM1's upregulated, there's innate immune activation, and we're characterizing all that now. And in the future, we'll be looking at the recruitment of envelope-specific B cells, whether we're increasing local concentrations of envelope-specific neutralizing antibody. We'll be enumerating our cell populations via imaging and moving on to further protection studies. But my basic point here that I want you to consider is, can CD8 T cells and humoral immunity work together? And maybe, again, this is the way the immune system is supposed to work, where you don't balkanize your different functions and that the two sides communicate with each other. And that makes more sense to me. But even if that's not true and they are separate, can we do a better job of leveraging both arms of the immune system more optimally in the vaccine space? And should we put more effort there? And I'll just briefly acknowledge the wonderful team at Emory. I've just highlighted the all-stars there. And then my lab, and particularly Vineet Jog, for driving most of the new work that I presented today. Thank you.
Thank you very much, David, for this fantastic presentation and how we have to look into the mucosa also to see what happens to protect from infection. Now I have the pleasure to introduce the next two speakers, two abstract-driven presentations. The first one by Federico Garcia on transmitted drug resistance to integrase-based first-line HIV antiretroviral treatment in the European cohort, the MeditRes HIV collaboration. And the second one by Kenji Nishiura from CDC on PK and PD relationships of vaginal applied integrase inhibitors in macaques. I want to remind you all, please put your questions on the Q&A, and this is the opportunity to have all your questions then answered during the Q&A session. Put it in the chat. Thank you.
Hello, everybody. It is my pleasure to present our data on integrase-based first-line HIV antiretroviral treatment in the MeditRes HIV collaboration. So as you may know, integrase strand-foot inhibitors-based regimens are preferred regimens for first-line antiretroviral therapy. INSTI baseline resistance is not recommended by current clinical guidelines. Currently, there is a growing interest on rapid initiation of first-line therapy. Dual therapy, DTG plus 3DC, is now recommended for first-line therapy by some clinical guidelines, and there is a great debate on whether transmitted drug resistance may hamper its use for test and treat strategies. So, of course, monitoring of transmitted drug resistance in real time is needed, and of course, we believe that it will bring some light into this very, very interesting debate. So in this study, we aim to evaluate the prevalence of transmitted drug resistance to the integrase and the NRTI backbone in newly diagnosed patients that are naive to ART. For this, we use MeditRes HIV, which is a consortium that includes ART-naive people living with HIV that have been newly diagnosed in France, Greece, Italy, Portugal, and Spain during years 2018 and 2019. So, in this collaboration, what we have done, we have sequenced the RT and the integrase following the standard methodologies that were in use at the participating centers in the study. To evaluate the prevalence of the surveillance drug resistance mutations, we used the calibrated population resistant tool for integrase and RT, which is available at the Stanford HIV website. And to evaluate clinically relevant transmitted resistance, we use the Stanford HIV database algorithm. Clinically relevant resistance was defined as any resistance level for Stanford interpretation that was equal or higher than 3, which, as you know, is low-level resistance. So we've been able to study 1,844 patients in which we had a paired RT and integrase sequence available. The median age of the patients was 40, 79% of the patients were male, 60% were men that had sex with men. The median logarithm of the viral load in copies per ml was 5.01, with quite a high proportion of patients with viral loads above 100,000 and almost a quarter of the patients having viral loads above 500,000 copies per ml. The median CD4 count was 330 and almost a third of the patients were below 200 when we included them at the study. Almost 50% of the patients were infected by non-B subtypes, and CRF-O2AG accounted for mostly all of a great number of patients that were infected with non-B subtypes. So let's see what we found when we looked at integrase-transmitted drug resistance. When we looked at the mutations that are included in the surveillance drug resistance mutation list for integrase in Stanford website, we just found 4 out of 1,844 patients with SDRNs. This is very low. It's just 0.22% of the patients. One of these patients harbored T66I, one T66A, one E138T, and interestingly, one patient harbored R263K, which you know limits the activity of second generation INSTIs, dolutegravir and bictegravir. When we looked at clinically relevant resistance, we found that 2.45% of the patients, which means 45 out of 1,844 patients, had any level of resistance using the Stanford HIV algorithm. If we looked at the data, we can see that just one patient had intermediate resistance to bictegravir and dolutegravir, which is very, very low. This was the patient that harbored R263K. And the rest of the patients were harboring intermediate or full resistance to the first generation integrase inhibitors, raltegravir and elvitegravir. And in the bottom of the slide, you can see which mutations were responsible for this level of resistance to the first line integrase inhibitors. So, INSTI-transmitted drug resistance in this cohort was very, very low, just having 0.05 of the patients with drug resistance to second generation INSTIs. When we looked at an RTI-transmitted drug resistance, we found 3.6% of the patients. This means 66 patients out of 1,844 patients with surveillance drug resistance mutations. Interestingly, 16 patients were harboring N184V, 2 were harboring K65R, and as you can see in the slide, most of the patients were harboring TANs, and these mutations were responsible for a great number of the patients that had SDRNs in the RT associated with NRTIs. When we looked at the clinically relevant resistance, we found that just 1.7% of the patients were harboring clinically relevant resistance to first-line NRTIs. This means that just 31 patients out of 1,844 patients were harboring clinically relevant resistance. And interestingly, less than 1% were harboring clinically relevant resistance to TDF-TAF and to 3DC-FTC, and 1.5% were harboring resistance to abacavir. So with this data, we would like to conclude that in this paper, we describe the most recent data on transmitted drug resistance to integrase-based first-line regimens in the Mediterranean Europe. We conclude that given the low prevalence of clinically relevant resistance to second-generation integrase inhibitors and to first-line NRTIs in the years 2018 and 2019, it is very unlikely that a newly diagnosed patient in the MeditRes countries would present with baseline resistance to a first-line regimen based on a second-generation integrase inhibitors. Less than 1% of the patients would have this resistance to the NRTI or to second-generation integrase inhibitors. So, with this data, we would also like to conclude that baseline resistance may not be an issue for rapid initiation of an INSTI first-line based regimen. And with this, we would like to stop, and first we would like to acknowledge the team from France, led by Anne-Geneviève Marceline, from Italy, led by Mariella Santoro and Francesca Ceccarini-Silverstein, all the centers that participated in CORIS from Spain, the team from Greece led by Dimitris Paraskevin, the team from Portugal led by Perpetua Gomez, and we also would like to acknowledge VIV for financial support through an unrestricted grant. So thank you very much, and I will be happy to take any questions. Thank you.
Hi, my name is Kenji Nishiura, and I'm a member of the laboratory branch in the Division of HIV/AIDS Prevention at the Centers for Disease Control. I'm here to share the results of a study looking at the pharmacokinetic and pharmacodynamic relationships of vaginally applied integrase inhibitors in macaques. Although only daily oral tenofovir-based products have been approved by the FDA for HIV prophylaxis so far, clinical trials have demonstrated that vaginally applied antiretrovirals can reduce HIV acquisition in women when adherence is high. Most notably, this was demonstrated in the CAPRISA-4 trial with a vaginal tenofovir gel and in the Aspire trial with the dapivirine vaginal ring, which was recently approved for use by the European Medicines Agency. Both products could prevent HIV infection if used before exposure, but the development of post-coital, post-exposure prophylaxis could provide additional options that give women more control over their own health. On the right is a modeled depiction of the temporal dynamics of steps in the viral replication cycle of HIV in a target cell. Reverse transcriptase inhibitors like tenofovir and dapivirine have a narrow window to accumulate to protective levels by the time reverse transcription begins. But because integration initiates several hours after target cells exposed to HIV, there's greater flexibility in dose timing that makes integrase inhibitors more suitable for PrEP. My colleagues have already conducted a study in macaques that has demonstrated this utility of integrase inhibitors for topical prophylaxis. Using a low-dose repeat shift challenge model, they showed that the integrase inhibitors raltegravir and L812 could be formulated in vaginal gels and respectively confer highly effective post-exposure and pre-exposure prophylaxis. However, the study did not assess the drug levels in mucosal tissues at the time of virus challenge, and pharmacokinetic correlates of protection were not determined. In the study I'm presenting today, we looked to fill that gap by retrospectively identifying concentrations of raltegravir and L812 associated with highly protective vaginal gel doses. In order to accomplish that, we designed our study in the following way. We evaluated the pharmacokinetics of raltegravir and L812 in cycling female pigtail macaques. Intravaginal dosing was repeated weekly over 4 consecutive weeks to account for potential variabilities in drug absorption over the course of the menstrual cycle. We then collected plasma, vaginal and rectal fluids, and vaginal biopsies at the indicated time points. First, let's look at the drug concentrations in plasma. Although many of the plasma samples in both raltegravir and L812 arms were below the limit of quantitation as indicated by the dotted line, we were able to detect their presence in plasma at the earliest sampled time point of 30 minutes, indicating that they're capable of rapidly penetrating from the vaginal lumen to the underlying basal environment. Generally, we found that there was low systemic absorption. Next, we'll look at the drug concentrations in vaginal fluids. We found that raltegravir and L812 drug concentrations in vaginal fluids were highest at 2 hours and could be durable over the course of 24 hours with concentrations 4 to 5 orders of magnitude above the protein-adjusted IC95. Next, we'll look at the drug concentrations in vaginal tissue biopsies. Mirroring the temporal kinetics in vaginal fluids, we found high and sustained raltegravir and L812 concentrations in vaginal tissue. This indicates that more than 200 micrograms of these products per gram of tissue may be needed for highly effective prophylaxis in vivo. We also observed that concentrations in vaginal tissue were well correlated with concentrations in matched vaginal fluid samples. We observed a strong positive correlation between drug concentrations in vaginal fluids and tissues, indicating that swab sampling alone could be a less invasive surrogate for tissue biopsies. Finally, let's take a look at drug concentrations in rectal fluid. We found that both raltegravir and L812 levels were high relative to the PAIC95 and showed potential for bicompartmental dosing of the rectum with intravaginal dosing. In summary, we identified the median mucosal raltegravir and L812 levels associated with in vivo protection. We observed that integrase inhibitor concentrations in vaginal fluids may need to be several orders of magnitude above the PAIC95 for highly effective prophylaxis in vivo. Products with integrase combined formulations for topical HIV PrEP and PEP. I'd like to acknowledge the following members of our branch for their respective roles on this project, and in particular, Dr. Charles Dupard for his time and his mentorship. Thank you.
Thank you very much. Again, really great presentation. My co-chair and I will be trying to look at all the questions that have come in and then try to gear those discussions with you all. Let me see first a question for Dr. Garcia regarding the low prevalence of INSTI mutations, transmitted INSTI mutations. What is the explanation for that? And how much INSTIs are now being used in the countries sampled?
Well, thank you very much for the question. Well, in the countries from especially Spain, Italy, and France, integrase have been used as first-line treatment for, I should say, more than 4 or 5 years. So, of course, one could expect that when the history of ARV treatment is that when you start to use a drug, you get transmitted drug resistance 2 or 3 years after the start has been introduced into the market. This is not the case for integrase inhibitors, and especially for second generation. And I'm pretty sure this is because of the high genetic barrier to resistance of first dolutegravir, who was the first second generation INSTI that we were able to use, and now bictegravir, who that is also being used. So of course it has a lot to do with the high barrier to resistance. We've seen this also with the PIs. When we started to use potent PIs and with a high barrier to resistance, we found that the failures to this resistance. So if you don't have resistance, patients failing with resistance, drug resistance. So I think this is the explanation for not having any transmitted drug resistance, especially to second generation INSTIs. Yeah,
definitely. I mean, this is all good news. And the data you mentioned actually mirror those from the US, exceedingly low level of transmitted resistance at this point. And it has to do with what you have mentioned, and in fact that patients are failing less on these regimens. They're well tolerated and so forth. So therefore, even the opportunities to generate resistance are lower than in previous regimens. And this is great news for integrase-based preventions like cabotegravir and whatever. So you don't have to deal with circulating resistance in the community. So. Great. Let me see, I can take on some maybe other question, and this is probably for...
Can I ask one question to Federico Garcia? I saw that you have collected sequences from 2018 and 2019, or patients from 2018 and 19, but not from 2020. Is that because there was a stop due to COVID? Is because you don't think it's any more worthwhile continuing, or funding problems?
No, it has to do with COVID and with time. Of course, COVID has stopped a great part of our research here in Europe, because as you in the US, you've been struck really, really hard by COVID. And but we used to do these studies. I mean, it's really difficult to find studies in real time for transmitted drug resistance. If you look into the literature or abstracts, you always find data from 2 or 3 years behind the year you're presenting. So we're trying to collect the data for 2020 this year. We start on January, and hopefully we will be able to have our results by March. So of course we are continuing our collaboration, and we'll continue. I think it's very, very needed to monitor transmitted drug resistance, because you need to know what's happening in real time.
Yeah.
Yeah.
Thank you. That's very, very important. Walid, you can go on.
All right, thank you. Thank you, Gabriela. So a question maybe for Gerardo. Is there any evidence for equivalent protection occurring in cisgender women for on-demand PrEP as opposed to MSM, considering the difference in vaginal and anal microbiome and immune reactivity?
Thank you. That's a great question. Unfortunately, we don't have any evidence of equivalent protection with on-demand PrEP in women. There are some questions regarding the differences in pharmacokinetics or drug penetration in vaginal tissues that might potentially affect the efficacy of on-demand PrEP in women. There is no data to support that, but we know that, for instance, tenofovir penetrates less efficiently the vaginal compartment compared to, for instance, the rectal compartment. So these are studies that need to be done. The question in the field is whether you will need higher adherence, you will need higher adherence in women than in men to be able to see the same level of protection. We do have some studies done in macaques where we evaluated 2 doses, one before and one after shift challenge. We found 100% protection of those 2 doses, whether the animals were, study was done as is, or the animals were receiving the injectable contraceptive, Depo-Provera. We saw some hints of a little bit of a reduced protection in the presence of other sexually transmitted infections by the 2-dose regimen. So maybe there is something there. It still was significant, the protection, but maybe there is something there that could potentially affect the efficacy from the PrEP in women. But definitely that's an area of research that we need, and we don't have the data yet in women.
Thank you, Gerardo. That was great. A question for Kenji. Your finding is very important, the need for several orders of magnitude of drug in the vaginal fluid to achieve protection. Do you have any explanation for that when you compare to the in vitro, for example, IC90 levels?
I think it's just messier in vivo. I mean, there's a lot of things that are going on and that don't take place in a cell culture plate. I think it's as straightforward as that, I guess.
Yeah, clearly. Drug diffusion and stuff, the difference in vivo than in vitro, and that probably would explain that. Let's see. Shall I take a couple more, Gabriela, or you want to practice?
We can come up. There is a few for Dave.
Go ahead.
Okay, shall I do it?
Sure.
Okay, Dave, there are 2 for you, and one is asking you if you or anyone looked at what happens to the CD4 TRM during CD8 TRM reactivation in tissues.
Sorry, I was muted. Yes, we have looked at that. There is a little bit of data in that Nature Medicine paper looking at as a consequence of CD8 T cell reactivation specifically, and we know that because we were using a peptide that would target the CD8 specific T cells. The CD4 positive TRM upregulate viral restriction factors in genes in response to basically interferon signaling. We've built on that much further, doing sort of in vivo CD8 TRM reactivation challenges, and then taking a site-seek approach and looking at really a number of fundamental changes in local immune populations that definitely include the CD4 T cells. If the spirit of your question is whether that would sort of fight against the possibility of increasing vulnerability of neighboring CD4 T cells to HIV or SIV infection, I think it's a great question. My bias based on the signatures that we're seeing is that the answer is no, but I think that is something we're very mindful of.
Thank you very much. There is another question for you, Dave, and that is more specific and says, were the animals used for prime boost strategy on hormonal contraception? And if not, did you know cycling status at the time of infection?
The answer is no, they were not on contraception. We did acquire hormonal or cycling data sort of for a post hoc analysis that I don't think was particularly revealing. If this, the protection data I really showed was a repeat challenge, low dose. So you're looking at, first of all, contemporized controls, but also challenges that occur over a relatively long period of time.
Thank you very much. I was very impressed by your data. I think it's exciting.
Yeah, very promising.
Yeah, promising to see that B and CD8 might work together. I was wondering, did you do some CD8 depletion experiments in the macaques?
We have not. I think that's a great idea and something we'd like to pursue. There are some caveats with CD8 depletion in macaques, but it's certainly something that is strongly being considered for the future.
So, you have all been talking about mucosal tissues, and especially for the drugs. I was so much impressed how fast these drugs are penetrating in the mucosal tissues. And so I want to be a little bit provocative. Yesterday, unfortunately, we didn't hear about the AMP trial results yet, but antibodies may work or may not be as efficient yet, or we don't know yet. But with all what you have shown, do we really need a new type of intervention? Or do we have already good enough approaches? You showed also incredible new promising regimens, promising new systems to prevent that could be used. So what do you think?
And who wants to field that?
So I think it all depends on the duration of protection. I think we have agents that we know can protect, and we have whether a daily pill protects. We know an injection of cabotegravir protects at least every 2 months. We don't have products at this point that could protect for more than 2 months, perhaps 6 months, 1 year. So whether it's going to be an ARV delivered for an implant, or it's going to be a monoclonal antibody, a blood-interaction monoclonal antibody, we don't have the information now. But I think definitely at this point of the game, yes, we have a few drugs that will move in, a few monoclonal antibodies. I think what we need now is to expand the level of protection and to deliver those particular ARVs or antibodies through different mechanisms. So I think there is definitely a room still for monoclonal antibodies.
Nobody else has opinions?
I mean, Gabriela, this is just to chime in. I think yesterday's one theme came out is that availability of different options, because different users may prefer different options for PrEP. And from that angle, you could kind of think of, some do well with a daily pill. Some struggle with that. They require a long acting. We're probably going to have soon a long acting for 2 months, like Gerardo was saying, but now the field is moving. Can we extend that? Can we have implants or injectables for longer than that? But then the other issue is topical. How much do we really need topical prophylaxis this time? And then the need for very high drug levels for the mucosal protection by topical products. So, and again, people argue that, some people prefer a topical application and insert on demand versus a pill that you take systemically or an injection or stuff like that. So people make often the comparison with contraception and the availability of different options for different groups. So
I was impressed by the fact that all the participants, or almost all the participants of the AMP trial, agreed and went on every 8 weeks to get
infusion
that was pretty long and demanding. So there is the will to continue on that front also.
Yeah.
I can imagine also that it would be better to have a shot that one can do once every 6 months and be subcutaneous, or something more easy to.
And there's the cost effectiveness angle too. How do how much is going to cost versus those newer products.
Yeah, drugs are possibly less expensive than not a monoclonal antibody.
Yeah.
So,
okay, any other questions coming in? Let me see so far.
there is a comment.
No, I mean, one question, I had some internet interruption, so I don't know if Gerardo, you have commented on that in your talk regarding the ability of the macaque model to predict the clinical efficacy so far. What are your thoughts on implications of this model for regulatory approval? How can we use those data to expedite approval in one way or another? It's just been on target predicting really clinical efficacy in men and women, at least from oral Truvada and TAF-FTC and Cabo, as you've indicated. So how do we start using those animal data in a really well-established, standardized model to kind of drive regulatory approval and expedite?
I guess that's the money question. So far has been, as you mentioned, very predictive with different products, whether they are oral or injections or different ARVs. So the predictive value appears to be there as long as the models incorporate a very robust PK component, so we can compare or we can model PK drug distribution in humans. And a relevant viral challenge component, I think we are getting there. I think it's a discussion that needs to happen to understand, okay, now that we know we've been predictive value for drugs from different classes and through different routes of delivery, what is the next step? Can we use this as a surrogate for protection? Again, this is a big discussion that needs to happen. I'm not sure if the field is still ready to say, let's use animal data to, let's say, if this is protective against rectal transmission, can we use animal data to demonstrating vaginal efficacy to perhaps move the intervention to woman? Definitely it's a big discussion that needs to happen, but we are not there yet.
Yeah, clearly the FDA has to weigh in, and but it's given the challenges of clinical trials, we're not having placebo anymore and having, Truvada or Descovy and the high cost of running those trials, and that's also includes vaccines as well and BNABs. So there should be some sort of a discussion on how do we maximize the utility of these predictive models for product approval.
Definitely. I mean, and now if cabotegravir gets approval, which we hope we get, that now the comparator arm will be an intervention that has shown to be extremely effective. So definitely that's going to be an important discussion in the setting of the next generation of clinical trials for drugs that we not prevent, but that can be delivered through different mechanisms like implants, intravaginal rings, or so we need to have that discussion. Absolutely.
Yeah. Thank you. Gabriela, anything at your end?
I've seen there are two comments that are referring to the discussion we had before by the same person that is saying that it would be better to have contraception might be good too. And then a comment that was, yes, we definitely need new and better PrEP, easier to take, fewer pills will dramatically expand group of people who might consider this. And
yeah, it's a important point. Yeah, more options, and the field is moving into non-daily dosing, either on demand or fixed once weekly or once monthly, and a pipeline of exciting also injectables and implants. So this is evolving very fast in a nice way. So
add that it would be nice also to have all the governments that play into this, because at least when it comes to Italy, it has been, PrEP has been approved only recently, and it's still not really covered by the public system. So
yeah, its cost is Truvada has become generic in the US, and Europe has been generic for a while, so that's gonna help with cost. So
very good point.
Yeah.
So I think there are no more questions from the audience. The last chance to speak virtually. It's unfortunately, it's not as fun as being all together, but this is a very good way to keep on great seminars, discussions, a little bit more tricky to get into these chat boxes and use, but no more questions. I think if there are no more questions, do you have some questions, Walid?
No, thank you.
So I think we can, are you all satisfied by the discussion? And I want to thank all the presenters. It was really a very nice, great data and great review that you gave on your data and also other data, on published data. And so I close this session and hand over to Walid for the closing.
Okay, thank you very much, Gabriela, and thank you again for session 4 presenters. At this point, we're reaching the end of the workshop. I'd like again to thank everyone, the organizing committee members, as well as all the presenters, virology education, for their support, and also the support of the funders that make this happen. Looking forward for next year, this year, as you know, has been exceptional. We're a little bit hopeful for next year, and the discussion within the organizing committee is right now, how do we plan for the meeting for 2021? We look like we're leaning towards a meeting in November, possibly at the NIH campus in Bethesda, with a possibility of being a hybrid in-person and virtual meeting. Nothing is set yet, but that's the discussion. We obviously will be sharing all that information with all of you. Again, a reminder to put in your evaluations for the different sessions. We definitely look at that, and that helps us plan for a better meeting for next year. And with all that, I'm happy to close this meeting, and hopefully looking forward for our next one at the end of 2021. Thank you, and bye-bye.